give folks just another second here to get settled, and then I'm going to get started. Some of you saw a little bit of this already at my table yesterday, and so one of the things that I might ask if somebody's feeling brave and camera ready this morning is if somebody would like to join me up here to help me kind of move the pieces along while I'm talking through the model. Anybody feeling like that is okay? Because I am also very capable of doing that. But I just thought it could be fun. If you got to see it yesterday, you don't need to. I mean, you can. I don't mind. I can help. Come on up here, Charlotte. Okay. So, Charlotte, what I'm going to do is sort of talk through the model. And as I'm talking through pieces, I'll then cue you to move some of these pieces as I'm describing the process of the model. Sound good? All right. Are we ready? Awesome. Okay, so to start, when we're talking about gene therapy, what we're really looking to do with gene therapy approaches to delivering medicines is answer, I think, a question that we've been curious about for a very long time in science. Can we use genetic material to treat genetic diseases, really kind of at their source? And this has been a field that's been in research for 50 plus years. So while I think sometimes this feels almost a little bit like science fiction, it's been around in science for a long time. And what I'm going to talk through today is what is adeno-associated virus and why do we use it? How do we approach delivering gene therapies for Huntington's disease, right? And what happens once it's delivered in your body? So let's start with what is AAV, because that's an acronym that you hear a lot when you hear about gene therapy, and what it stands for is adeno-associated virus. And we really like adeno-associated virus for a couple of really critical reasons, and this is our little model of what that looks like. Okay, adeno-associated virus, couple of really key features. One, it's a virus, and while that can sound scary on its surface, we really like them because they evolved with us for hundreds of thousands of years to be very, very good at their job of pushing genetic material into the cells, right, of our bodies and lots of other animal bodies. That's a mechanism that we're going to harness in gene therapy. So that's why we use a virus to begin with. The other reason we like adeno-associated virus is the associated part. So oftentimes viruses, because they're real amazing in a lot of ways, can replicate themselves. And adeno-associated virus cannot, because associated in this case means it has to have a companion virus around in order to do that work. And so we know when we're using adeno-associated virus that we have a lot of control over dose, we have a lot of control around what might be happening once it's in your body in ways that with other viruses we might not. And then finally, we really like adeno-associated virus because we've known about it for a very long time, and we know that it does not cause disease generally in humans. So it's a benign virus to our bodies. Lots of us have already been exposed to AAV often without ever knowing it. And so for those three reasons, right, that viruses are a great mechanism in terms of how they've evolved, that adeno-associated virus means that it can't replicate on its own, and also that it doesn't cause disease in people, it made AAV a really great candidate. And so a lot of times when you're talking about gene therapy, you'll hear AAV as the platform or the way that they're delivering the medicine. So I just wanted to make sure we covered off on a little bit of why first. Okay, so Charlotte, the first thing that anybody does when they're looking at developing a gene therapy is first, while we love the mechanism of this, we don't want that viral DNA in there anymore, do we? No, that's not going to help sort of the whole process. So the first thing we're going to do is we're going to remove that genetic material. So you can take the little lid off. Yep. And then you can just go ahead and set that down for a second and pull out that viral DNA. So in the lab, this process is sort of a process of washing where we're removing the viral DNA, and you can just set that aside. So that what we're left with, and then if you want to hold up both those pieces for me, both these pieces, sorry. What we're left with is cargo van, a little delivery vehicle that has been emptied out of its original contents so that now it's ready to receive whatever kind of cargo we might want to put inside it. Then we come into our labs and we build the cargo that we're looking to deliver. And in this case, that's the genetic material that I referenced at the beginning. So this is a piece of DNA, and DNA is, as you heard this morning, like the recipe book. And it gives the instructions to messenger RNA to go out and make all of those various proteins. So in Huntington's disease, we're building a DNA that will often go after that messenger RNA, typically using a little RNA that it makes. And so that's how this piece of cargo will work. And we'll explain more of that as we talk about what happens when it gets into your cells. But for now, Charlotte, if you want to put that piece of DNA in our little cargo van and go ahead and close it up. This is now what we would call a vector. And you'll hear that word a lot in gene therapy because now we have a fully complete packaged therapy ready to be delivered. We've got a piece of DNA that is the cargo we want inside a mechanism of delivery that is just real good at its job. Like I said, the viral shell. And that is the vector. Now, we need to deliver it into, in Huntington's disease, what parts of the brain? Pop quiz. Anybody feel brave? What parts of the brain we're trying to get to? There are two pieces. Yep, the caudate and the putamen. Exactly. So we're trying to get this therapeutic to the caudate and the putamen. And I just want to show you in the brain model where that sits. So I've opened this up so that you can see we're right in the center. When you hear folks talk about these deep brain regions we're trying to get to, literally, right smack dab in the deep middle part. And we have your putamen, which is sort of the plump little structure in the center. That's how I remember, deeper putamen. And then your caudate, which comes around that and has that kind of funny little tail, right? These are the two structures that we need to get this vector to. And so the way in gene therapy currently that we do that is through something called intraparenchymal infusion. Don't worry, I'm going to explain what that is, right? Intraparenchymal infusion is a surgery, so you'll often hear that these are delivered through brain surgery. And Charlotte, can I hold this up just so people can see it? We use what's called a cannula. Okay, it's a very thin, and you're welcome to come up after I'm done presenting. I'd love for you to come up and look at some of these things. A very thin needle with a guiding element. And a neurosurgeon will use an MRI machine and this system while you are under general anesthesia to get this vector down into these deep brain regions. Okay? Are there any questions so far? We're all good. Okay. So, you can go ahead and put that down. Thank you so much. So once that's been delivered, what happens? Okay, so this is a cell model. You want to hold that up so they can see? And not a lot of detail here, but the blue part essentially is the nucleus in this cell, right? And then you have all of this sort of cellular space out here where there's a lot of trafficking going on of RNAs going out to make proteins and coming back, stuff's coming back in. But we're going to talk mostly about the nucleus for right now. So once this comes into your body, it's going to come up to a cell in your brain, and it's going to do what we love about viruses in this sense, right? What's it going to do? It's just going to push that right in there. Can I stick that in there for me? You're doing great. This just becomes a waste product. Your body doesn't need the delivery vehicle anymore, and this is just going to get flushed out over the next couple of weeks from your body when you go to the bathroom, most likely. Now, sitting inside your cell nucleus is this little piece of DNA. And I want to be clear that in gene therapy, right, we're not talking about gene editing, those scissors. This is not going to change your DNA. Your DNA stays the same. But this will sit alongside your DNA in the cell nucleus and will create what we call an episome. It's just a little self-regulating, separated piece of DNA that unravels and kind of makes a little Ferris wheel of sorts. It's like a little circular structure that forms, and it starts broadcasting out a recipe. And in this case, in Huntington's disease, that's often a recipe for an RNA. So that RNA can then go and find your messenger RNA for Huntington protein and bind onto it and degrade it, which basically means break it down, shut it off, so that you will make overall less Huntington protein. Now, in this approach that I just described, you're going to make overall less Huntington protein of both the wild type and the mutant type. But the hope in gene therapy research for Huntington's disease is that if we're lowering Huntington overall, we're also lowering enough mutant Huntington in these really critical structures that we're going to get to some meaningful effect in hopefully slowing down and maybe even addressing some symptoms. But that's what we're learning right now in studies, right? And so we don't know yet we have some indication, but we're still, right, I want to be clear, learning because we're in clinical research. But that is essentially how it works. So I'm going to stop here and now encourage you, if you are comfortable, to come up out of your seats if you want to look at some of these things. And now we can also start the questions and answers, because I really wanted to leave a lot of time for just a discussion.